Okay, hello. Hello, guys. We'll continue with the non-coding RNA. Today, we'll talk more on the small non-coding RNAs. That is, this group of regulatory non-coding RNAs. So basically, we'll talk more about microRNA, siRNA, and piRNA. And more, just mention briefly about the Crashy RNA and TELS RNA. These are very new class of small non-coding RNAs. So to go ahead, we talked last time the long non-coding RNA, how those are generated, uh, more of the intronic RNAs, enhancer RNAs, promoter RNAs, all those things have been we have talked about. So today, to go ahead, we start with the uh, small RNA, more importantly the microRNA and the shRNA, and, or we have we'll be talking briefly about siRNA. Okay, so in microRNA, as we talked about, small RNAs are around 20 base pairs long, and most of the microRNAs are around 22 nucleotides in length. And there are three ways that you can get the microRNA. One is the classical or canonical microRNA, wherein you get a transcript which is longer than the mature microRNA, which is called as a primary microRNA. And this primary microRNA is processed by enzymes, something like endonuclease. In this case, if you look at here in Drosophila, mostly it's from Drosophila. So one of the RNA binding protein DZCR8 binds to the primary microRNA, or generally recognized the loop structure here, or the loops which are present in primary microRNA. And this binds and recruits the endonuclease DROSA. And DROSA, along with DROSA, allows the generation cleavage of the primary microRNA to give the pre-microRNA. The pre-microRNA is transported out of the nucleus in, as we talked about in earlier also, it's in a exportin 5 mediated RAN CTP mediated pathway. So exportin 5 binds the pre-mRNA and comes out of the nuclear pores, and this, once you get these pre-microRNA in the cytoplasm, then this is bound by another endonuclease, or endonuclease called dicer. This dicer complex is a complex which has endonuclease activity, and this cleaves the loop structure which is present in the pre-microRNA, and also and once this is cleaved and the loop structure is out of this pre-microRNA, then you can get this double-stranded RNA by recognized by the microRNA RISC complex. Once you get the microRNA RISC complex, it has various roles and which can act in different way. We will discuss it in the later part of our discussion. So basically, this is the canonical part that we know of, and you can get microRNAs from different ways, from the stem loop hairpin structures. In a normal mRNA, many of times it can come from the intronic region of the mRNA as well. These stem loop structures or short hairpin structures are recognized again by the same DZCR8 and DROSA complex, and which is exported just like in the canonical primary pre-microRNA, it is exported out in export in 5 dependent way. And another way is that you can get, as we talked earlier also, there is another way which can come from the M7G pre-microRNA. In this case, many of them, what you can see is that in a canonical pathway, it's generally, it can be coming from the polymerase, RNA polymerase 3 transcript. And this is the canonical pathway. But in the non-canonical, it can also come from the RNA polymerase 2 mediated transcript. So in this case, you will have M7G cap, and these structures are exported out depending on the recognition of these microRNA structure through exportin 1 and comes out of the nucleus. Once it is got out, it can be processed by dicer in the case of M7G cap pre-microRNA. But in the case of the shRNA, you associate with AGO, Argonaute complex, rather than the dicer. And and finally, it integrates all of these microRNA at the microRNA RISC complex. Okay, so this is about the biogenesis of microRNA. MicroRNA can be originated from RNA polymerase 3 mediated transcripts, that is the canonical pathway. And in the non-canonical pathways, you can get from the intronic regions, or it can be coming out of the RNA polymerase 2 mediated transcripts. Okay, once you get this microRNA RISC complex, it can go into different ways. Before we go into that, there is another class with of small RNA that is called the short interfering RNAs, siRNAs. So this is more or less lengthwise is very much similar. Many times the difference is what you can see is that the microRNAs are encoded in the endogenous genes of the genome, but siRNA generally is from the virus encoded or repetitive element encoded or transposon mediated, transposon originated from transposons. So generally you can get single-stranded again from the stem loop structure from single-stranded RNA. But in the case of siRNA, the transcript from where siRNA is derived, it is generally derived from the double-stranded RNA. What do I mean by this is that in the case of microRNA, you get a single transcript from a gene, and then it is processed to give rise to microRNA. Of course, during the process, we have base pairing, and you have structures formed, double-stranded structures are formed, but it is coming out of the single transcript. Okay, so to start out is a single-stranded RNA with stem loop structure. But in the case of siRNA, many times what it has been observed is that the multiple transcript from the single locus from both directions, or convergent transcription happens. So that means the transcription happens from the 5' site of anyone, any question? Or okay, so in the case of siRNA, we'll discuss later, it is generally because of the convergent transcription, means the transcription is happening from this side also, and transcription from this side. So both of the RNA forms a complementary double-stranded 2 fragments, and then it is processed further by the enzymes and go to the target site. And third difference is that one is the microRNA targets the gene not in a fully complementary way. or 1-to-1 base pairing doesn't happen. There is partial complement. That means some of the base pairs base pairing is there. In the case of siRNA, it's a complete match of the RNA, siRNA sequence and the target site. Okay, so there are few differences between siRNA and microRNA: the origin, what kind of genes they are associated, and then the kind of RNA molecule. Generally, microRNA comes out of a single transcript. siRNA many times is a double-stranded, means two strands are there complementing each other, and it's processed to give rise to siRNA. But the exon many times matches together, microRNA and siRNA. Both of them aid in the towards the cleavage of mRNA, so that there is no gene expression of the particular target. So to give into more into this aspect of microRNA biogenesis, we talked about DROSA, which is involved in the first step from the processing of primary microRNA to pre-microRNA step. What it does is that, along with the factor that is DGCR8, which is the RNA binding protein, it comes and interacts with this kind of stem loop structure and process and cleaves at the both the ends 5' as well as 3' end of the double-stranded structure, so that it gives a primary microRNA. Okay, so this is DROSA is RNase 3 enzyme. It's an endonuclease, and it has two domains of RNase 3 domains, and it cleaves. So it gives rise to stem structure hairpin flanking the hairpin loop structure. And we had another RNase which is involved in processing the pre-microRNA, pre-miRNA in the cytoplasmic site. That is done by the dicer. And as you can see, dicer is a RNA binding protein with the OB fold at the PAG motif in this or domain in this protein dicer. It comes and binds the microRNA or pre-microRNA at the 3', 5' free ends, and that gives rise to there is a conformational change with the metallic cation involvement, and it matures the pre-microRNA into microRNA. The story goes back, the discovery of microRNA goes back quite long ago. The first microRNA that has been discovered is the Lin4 microRNA, which was discovered in Drosophila system, Drosophila system. So what it has been known is that if you delete Lin4, the gene encoding Lin4 mutant had developmental defects. Basically, some of the I think there is egg laying capacity, or the egg laying was delayed when you delete Lin4. And Lin4 was not known in the starting phase in Horowitz, I think, Horowitz lab. So there they discovered the mutant in gene Lin4, but further on it was dissected by Ambrose and one of his colleagues to give rise to the phenomenon of discovering this microRNA. What the story goes is that when you have a mutation in Lin4, there is a maturation defect, thereby leading to delay in the egg laying process of the oh, sorry, this one was in C. elegans, not in Drosophila, sorry. This was in C. elegans. So the maturation of the C. elegans was delayed, and egg laying was delayed. But there is another locus called Lin4 lin4, when lin14, when you delete it, it is the of the opposite of lin4 activity. In other way, there is a pre or early maturation of the worms and premature egg-laying phenotype. So this was in trick, and then Ambrose et al. Looked at it. Ambrose and his colleague looked into this phenotype, and they found out that lin4 doesn't code for any protein, and it codes for RNA. So this then, because the lin4 mutation and lin14 mutation have opposing effects, they thought this lin4 might be a negative regulator of lin14. Okay, so before we go into the next discussion, we'll just briefly summarize what we have talked about. So microRNAs are small single-stranded forms of RNA, and these are encoded by endogenous genes and has hairpin shaped structures, and mostly it negatively regulates protein coding genes. And this number is very outdated one, it's a quite old one. So there are thousands of microRNAs in the human genome, and many of their effects are being studied. And most of the protein coding genes are either regulated by small microRNAs at certain level or long non-coding RNAs, and there are a lot of proteins being regulated by these microRNAs, small RNAs. So coming back to lin4 study, what Ambrose and others have found out is that lin4 doesn't code for a protein, but it codes for an RNA, and somehow this RNA goes and inhibits the expression of target mRNA, including lin14. So when they look into what it has been seen is that in the lin14 3' UTR region, that is untranslated region of the lin14 mRNA, there are multiple sites complementary to lin4 RNA. So that's how it has come up that lin4 is a negative regulator of lin14. So now, once they discovered lin4 and lin14 relationship, people looked into multiple other microRNA. One of the famous one is the LAT7 RNA. LAT7 again is involved in the developmental timing. So when they look at the expression level of microRNA and protein expression level, what it has been seen is that the lin4 RNA, when it is high, lin4, when it picks up, it knocks down the lin14 expression, including lin28 expression goes down. And LAT7 RNA, when it goes up, the lin41 protein expression goes down. So once this lin41 goes down, what happens is that another protein, lin21, comes up. So there is a very nice regulation of protein expression through microRNA. At certain stage of the development, certain microRNAs allows the transition of the stage by inhibiting the protein expression of certain proteins, like lin14 is regulated by lin4, and lin41 is regulated by LAT7, and giving rise to an adult stage which is marked by the expression of lin29 protein. So you can say in the larval stage of the development L1 stage, there is a transition of lin4 expression from the negative side, then it goes down by LAT7. So there is a very nice regulation from microRNA. Now the question is how this regulation, the negative regulation of expression by microRNA is done. So people have looked into the regulatory pathway, what it has been seen is that mostly if you microRNA, as we talked about, DROSA and DISA, along with the interacting proteins process to mature microRNA. Once it goes, RISC complex, the single-stranded microRNA, goes and binds to the 3' UTR of the target mRNA, and this RISC complex leads to the degradation of the target mRNA. Okay, RNAi pathway, just like the siRNA from the transposon or hairpin structures we do in the lab experimentation, what we get is double-stranded RNA from transposon, etc. Goes from double-stranded RNA, DICE recognize this and chops up into siRNA that goes into RISC complex and targets the particular mRNA. It can be targeting any part of the region. It can be at a 3' UTR or in the coding region. Once it is bind, it activates the RNA's activity and cleaves the target mRNA. So the question is how this cleavage by microRNA to inhibit the cleavage is done is that in the microRNA RISC complex, you have AGO protein and RNA binding proteins, binding domains. So AGO protein, which is there, finds the complementary region in the mRNA and also interacts with other proteins, which recruits certain factors like ZW182, which recruits other factors which will be important for the cleavage and recruitment of or degradation of the target mRNA. So how that is done is in the stepwise manner. The microRNA, as we have seen here, the complex AGO, ABD, ZW182 recruits certain factors, and then these factors in turn recruit other factors. So the stepwise manner, what it does is that AGO protein recruits along with ZW182 recruits PAN2 and PAN3, which results in the deadenylation of the poly-A tail of mRNA, of the target mRNA. So this leads to the deadenylation of the poly-A tail to a certain extent, and then recruits other factors like CAF1, NOT complex, CCR4, this kind of complex, and completely deadenylates to less than or around 10 poly-A tail. Okay, once it reaches certain states, it recruits this complex, the AGO complex also recruits the factor with something including the DCP1 complex that decaps the 5' G7 cap of the mRNA. Now, once deadenylation and decapping is done, both the ends of the mRNA are free, then RNA exonucleases can act very fast on this transcript, and this transcript is degraded very fast by the exonuclease activity of other exonuclease present in the system. So the steps involved in microRNA mediated target mRNA degrade is first you deadenylate the poly-A tail of the target mRNA, then followed by decapping of the M7G cap, and that leads to free 5' and 3' ends of the mRNA, which is accessible to ribonucleases, which are very much abundant, and that degrades the target mRNA. Okay, so so far the first question from the starting from the Lin4 and Lin14 discovery, what it has been seen is that the microRNA generally is seen to be involved in degradation of target mRNA, thereby leading to downregulation of the expression of particular protein or gene. Okay, so now there has been some observation in certain cases wherein the microRNA helps in stabilizing and allow translation of proteins. For example, in one of the studies in recently, what it has been shown is that in to normal uh, fast dividing cells, the microRNAs can recruit AGO or that microRNA risk complex and degrades mRNA and inhibits or downregulates translation. But when the cell goes into quiescent state or non-dividing state, many a times what it happens is it recruits the AGO to the microRNA risk component, recruits some factors like FXR1 that doesn't allow degradation of mRNA. Rather, it stabilizes the RNA and increases the translation of the target mRNA. So, microRNAs can be involved in shutting down the expression of a particular gene, just like in the classical observation of LIN4 and LIN14. But in other cases, when the cell shifts into certain mode from something like from the fast dividing mode to the quiescent state, there is a shift in the role of the microRNA risk complex, which can, rather than decreasing the translation of a protein, it increases the translation of a transcript. This has been observed very recently, around 2010. Okay, so another way the microRNAs is not only involved in mRNA stability, but it can also be involved in changing the transcriptional state of a certain gene. So the microRNAs has been observed in nucleus, and it's also known to change the transcriptional status of a gene. This can go in three ways. One is the microRNA and the microRNA risk complex interacts with a genomic region through complementarity and recruits transcriptional factors or histone modifiers, thereby changing the transcription of a nearby gene. And here TSS is the transcriptional start site. So by recruiting transcriptional factors, either inhibitors or activators, most of the time it's the inactivators, but it can also recruit histone modifiers and change the transcriptional profile. Another way is that the microRNA risk complex interacts directly with a nascent mRNA, thereby after interacting with this nascent RNA, it can recruit histone modifiers and maybe shut down or activate the transcription of the nearby gene. Third thing, rather than directly complementarity to the genomic gain and change and recruit transcriptional factors or histone modifiers, it can also go and form a complex with the target site in such a way that it changes the structure. Rather than doublet, it can change to quadruplex structure, or the structure of that genomic region is changed. So by changing the topology of the chromatin structure or the genomic site, it can change the transcriptional status. Okay, it can activate or it can inhibit the expression of that particular gene. So so far what we have talked about is that microRNA can mediate the degradation of a particular target mRNA. That is the classical first observation people have seen. The second thing is that it can change from the degradation to activation of translation. And fourth is microRNAs. Third is microRNAs can enter the nucleus and change the transcriptional status of a particular gene by three different mechanisms. So now, in the broader picture, what?
Excuse me, sir.
Yes, tell me.
Sir, in the previous slide,
Just one.
Yes, sir. In B, what is happening?
So in this B, it's not interacting with DNA. It's interacting with a nascent RNA which is coming out of RNA polymerase. So A and B is interacting with the genomic DNA segment, correct? But in the case of
Yeah.
B, it's interacting with a nascent RNA and helps in the localization of histone modifiers. So it's kind of indirect, correct? So it's slightly different mechanism.
Okay.
Yeah, is it
Yes, sir.
fine? Is it fine?
Yes, sir.
Okay, then let's go ahead. Huh? So now coming back to in the broader picture, what we have been talking about is the microRNA can be in the cytoplasm or in the nucleus doing different roles. But what it has been shown is that this kind of regulatory mechanism can be localized in different organelles or different location of the cell. For example, the translational regulation can happen on rough and endoplasmic reticulum. Rough endoplasmic reticulum is, as you all know, that it looks rough nature because a lot of ribosomes are at the outer membrane of the endoplasmic reticulum through uh, which helps in the translation of proteins which are supposed to be membrane targeting or to be secreted. So it can be acting at the translational start sites, thereby changing the dynamics of protein translation at the ER membrane. Or many a times, what it does is that it can associate with RNA and targets to certain stages like the lysosome. Lysosome, as you all know, the lysosome is the site wherein proteins, RNA, all these are getting degraded. And one more thing people have observed is that in the export vehicles or the small vesicles which are secreted out from the cell to the, for example, in humans, like extracellularly secreted vesicles are there, which can carry lot of these microRNA. And these microRNAs can go in these extracellular vesicles to different site and regulate the gene expression in that particular tissue. So now microRNAs can act extracellularly uh, through the exosomes, that is the extracellular uh, vesicles which are secreted out. And it can go to different tissues. It need not be at the same cell where it is synthesized. So now the question is, people have extended this observation. What people have done is, because exosomes are immunologically inert, so people have applied, what people have been doing is that try to overexpress microRNA in certain cell line, enhances the formation of exosomes and collect the exosomes from that preparation and inject into individuals in during particular disease or medical condition. That is one way to treat that the medical conditions people have been exploring. So if you look for exosome synthesis, all these things, microRNA becomes very important factor to regulate gene expression through exosome utilization. So microRNAs can be intracellular or extracellular. It can be near or far in its activity. Okay, so then next we go ahead into its sister uh, RNA, that is siRNA. As we talked about earlier, the siRNA comes out from the generally from transposon encoded transcript or from viruses, and mostly it is derived from the double-stranded RNA. Okay, so this double-stranded RNA, just the dicer which is involved in the maturation of microRNA, is involved, it recognizes and processes it. Once it is bound with AGO2 or Argonaut, which is involved in the microRNA risk complex. Argonaut, there are different types of Argonauts, and it changes the specificity of the target or what kind of microRNAs are going to be in that complex, and its activity is changed. But here, what I'm saying is that these siRNA after being processed by dicer, it binds to the RISC complex, and RISC complex in the RISC complex, what is done is that this double-stranded RNA, just like the microRNA, one of the strand is going to be degraded, and this degradation of particular strand it depends on the uh, GC content or what kind of ends are there, depending on that there are different criteria, but we need not go into much detail in there. So in the RISC complex, one of the strand of the double-stranded RNA, just like microRNA, it's degraded, and once that is degraded, it goes and binds to target site or target mRNA, and with full complementarity, unlike in the microRNA, which can be partial complementarity, uh, it can go and binds and degrades the or cleaves the target uh, mRNA. And what is important here is that this once the cleavage of the target mRNA is done, the RISC complex along with the siRNA, single-stranded RNA, can be recycled. In one way, what it means is that single microRNA or siRNA can do this cycling and degrades multiple copies of the target mRNA. So these are very efficient way to stop expression of a particular gene. Okay, now this is pretty much of siRNA. The processing transcription, as I told you, it was pretty much there. I think, let me check. Yeah. Something I'm missing here. Okay. So,
Sir,
yeah, yes.
there's a question in the comments. If you
Yeah, can you just read out because I'm in full mode. I cannot see. Yeah, what
uh,
is?
why is it called passenger strand? Just because it gets cleaved and thrown away?
Yeah, so it's kind of both strands are required to be recognized by this complex. So you are getting this kind of a passenger to carry this guide RNA. So functionally, we don't know how much relevant is this. Okay, so this strand doesn't have any biological significance. So it's just to get the guide strand for its biological function. Correct? That's why I think it is called passenger RNA. Yeah.
Yeah, thanks.
Yeah. So how that double-stranded RNA moves in the case of siRNA is pretty much same like the PV interacting RNA, piRNA. What it does is that, as I was telling you, the convergent transcription. What does mean by convergent transcription is that there is a locus, and RNA transcription can happen from both the directions. So what would happen is that if it is transcriptionally in a bidirectional or dual strand cluster, what it does is that it forms a double-stranded RNA that is being transported out of the nucleus, and that is processed by the dicer to give rise to that mature siRNA. So very relative one is piRNA, PV interacting RNA. This was first observed or extensively studied in Drosophila melanogaster. What it does is that PV interacting RNA is very important in down-regulating the expression of transposomal elements or repeat uh, elements. So in this case, what it does is that there is in the case of Drosophila, there is a protein called rhino. Okay, rhino. recruits factors and changes uh, the epigenetic of that particular locus. And, more importantly, H3K9 trimethylation, H3K9 trimethylation, and this allows the recruitment of RNA polymerase from either side because both the strands are going to be modified equally. So it allows transcription from both sides. What is very important here is that in both the cases of microRNA, there is a proper promoter, or even in the siRNA, there is a uh, very specific promoter and very specific start site for its primary transcripts. But in the case of PUA interacting RNAs, it just modifies and recruits, because of this modification, recruits the transcriptional factors, and there is no particular very specific site of transcription start site. So it's kind of random transcription bidirectionally. Once you get the transcript, this is processed by uh, enzymes or RNA binding factors, in this case DEL and CAF. In the case of Drosophila, it binds this double-stranded kind of RNA from the bidirectional transcription. And once these transcript recruits, UAP56 is kind of exporting, which allows the export of these transcript to the cytoplasm. And in the cytoplasm, it is processed by different complex. In the case of Drosophila, one of the main important enzymes called zucchini. Zucchini is mitochondrial localized, and it recruits other factors in such a way that it allows the maturation of these uh, uh, primary PUA RNA to maturation. And the mature state is bound by the PUA interacting proteins, AUB or PUA interacting proteins, and this goes and targets the uh, specific RNA or genomic locus that we'll discuss. What is important here is that which has come about is that in the case of PUA interacting RNA, the PUA interacting RNA complex, there is one, and another Argonaute proteins, Argonaute 3, can also be involved in the activity of PUA RNA, piRNA. So in this case, what is the difference between this PUA complex and AGO3 complex is that this RNA which is in the complex, they are more, generally complementary, and the PUA interacting is most of the time is antisense to the target RNA, but AGO3 is sense to the target RNA. So this has AGO3 complex has the same sequence as the target RNA, and in the case of PUA complex, it has complementary to the target RNA. But what is the importance of these AGO3 complex, which has complementary RNA sequence? It is seen that the number and maturation of PUA RNA is reduced when you knock out AGO3, indicating that AGO3 may be helping to find the primary piRNA to be processed by the processing enzymes like zucchini, and all is recruited at a particular primary piRNA sequence. So basically, it helps in the processing of the primary PUA RNA, piRNA, to the matured PUA complex. So that is the belief, but it is not shown very directly how well it is processed and how it is done. But it is seen that if you delete or muted AGO3, there is a downregulation in the maturation of PUA RNA complex. Okay, and more importantly, what it is seen is that PUA interacting RNA by RNA mechanism is very much conserved. You can see in mouse, C. elegans, and in the case of Drosophila melanogaster, it's more in the germ-line cells. So it's a tissue-specific activity is seen in the case of higher eukaryotes. Okay, now going to how PUA interacting RNAs acts on to shut down the transcription of transposons or repetitive elements are in two ways. In the case of Drosophila melanogaster, what it has been seen is that it can act at the transcriptional gene silencing or post-transcriptional gene silencing. So in the case of PUA for post-transcriptional gene silencing, it's pretty much like the microRNA mediated gene silencing. It goes and binds to the complementary sequence in the complementary sequence in the target RNA, and it is cleaved and down-regulates or degrades the transcript from the transposons or repetitive elements. But at a transcriptional level, as we talk about, just like in the microRNA, PUA interacting RNAs goes and binds to the nascent transcript and regulates or recruits transcriptional histone modifying enzymes so that it can shut down the transcription of that gene locus. So PUA by RNA can go and shut down the locus. So it's a feedback loop system in both ways. If it is transcribed, it is degraded in the cytoplasm. But in the nucleus, it can, if there is a transcription, it goes and binds to the nascent RNA and modifies the locus epigenetically and shuts down the transcription of transcription from that particular locus. That is one way. Second thing is it can also recruit this PUA microRNA once it localizes. It can recruit certain factors which can shut down the transcription of these repetitive element transcription or transposon related transcription. So this mechanism has been seen in mouse. It can help in post-translational knockdown, but most of the time it is at the transcriptional level. So it goes and recruits DNA methyltransferases. It methylates the locus by interacting with the nascent transcript and shuts down the transcription from that nucleus. So this has been seen in all different strata of evolution, that is Drosophila, mouse, C. elegans. You can see this mechanism happening. And other two small microRNAs is not well studied, and one of them is the centromeric repeat specific small RNA, CRASH-C RNA. This is discovered in the marsupial called Tama-Wallaby. It's not been observed so well in the case of other organisms. The first discovered in this marsupial, and what it does is that it inhibits the transcription from centromeric regions, and probably it is helping in the compaction of the centromeric repeats as well. That is what is believed. And another very recent finding is the telomere specific small RNA, and these are called TEL-S RNA. TEL from telomere, TEL hyphen small RNA is sRNA, TELS RNA. So these are transcripts coming from the telomeric repeats. And, basically, it's inhibits the transcription from telomeric repeats. So with this, I think we pretty much covered the small RNA non-coding regulatory RNAs. And then in the next class, I think we'll maybe tomorrow we'll discuss about some of the regulation, post-transcriptional regulation or transcriptional regulation, including this RNA-based regulation, some of them with examples. And then I think in the following week, we'll do that question discussion as we discussed earlier. Okay, and the videos of this class plus the slides will be shared. For video, I will be uploading in YouTube and share you the link. I think that may be easier because when I look at the size of the video, it comes around 135 to around the first one is 133 MB, so that may not be very good to send you as a file. So I'll upload in YouTube. I need to do some work on that because YouTube initially doesn't allow video more than 15 minutes unless you verify your account through mobile phone or something, then only you can upload that more than 15 minutes. So I'll finalize that and then upload you by this weekend. Okay, so then let me know if there is any question from your side we can discuss. Any questions? Mm-hmm. Then I'll stop for today here. See you in the next class.
Bye, sir.
Yeah.
Thank you, sir.
So there is one question there.
Yeah, tell me.
What was the function of? I can't read it.
The Cressy RNA. Cressy RNA is the centromeric repeats. Centromeres, you know, where you attach your microtubules and important for segregation of chromosomes. So these small RNAs inhibits the transcription from these centromeric repeats. So it's not well known because it's not well studied, and it has been observed in one of that marsupial that I talked to you. In humans and all, I think it's not well studied so far. Yeah, so there is not much information about Cressy RNA, but it is involved with centromeric repeats.
Okay, sir.
Yeah. Okay. Then see you guys.